All right, buckle up, everyone, because today we're diving deep into the fascinating world of biology. We're going to be cracking open the textbook, specifically Chapter 2, to really explore all those amazing mechanisms that make life tick.
Yeah, think DNA, genes, proteins,
Yeah.
all that stuff that really makes you, well, you.
It's like a backstage pass to the most incredible show, right?
Absolutely. It's the intricate workings of our very own cells.
Okay, now I know this might sound a little bit dense, but trust me, we're going to break it down into, manageable pieces and highlight the most interesting stuff.
Absolutely.
Think of it as a personalized biology lesson, just for you.
Yeah, I love that.
Have you ever wondered how like a tiny change in your DNA can actually has huge effects? Well, get ready to uncover that and so much more.
We're going to uncover all of that. Let's start with the absolute fundamentals, DNA, often called the blueprint of life.
Right.
But what exactly is this blueprint made of?
Yeah, it's a term we hear all the time, but maybe don't fully grasp.
So imagine a twisted ladder. That's your DNA, a double helix.
Okay.
Each rung of this ladder is made up of a pair of nucleotides, the building blocks, each with a sugar, a phosphate, and one of four bases, A, T, C, or G.
Okay, so A, T, C, G. So are these letters just arranged randomly or
That's a great
is
question.
there an order to it?
It's not random. The pairing is very specific. A always pairs with T, and C always pairs with G.
Okay.
This is called complementary base pairing.
Got it.
And it's super crucial because it allows DNA to replicate, to make exact copies of itself.
Okay.
And that's important because every time your cells divide, they need a fresh copy of that DNA blueprint.
That makes sense. So it's like each half of the ladder can be used to kind of like rebuild the missing half. But how does this ladder actually get copied? I can't imagine it's a simple process.
You're right, it's not. It's actually quite complex and elegant. It involves a team of molecular players.
Okay.
Picture this. An enzyme called helicase unzips the double helix, breaking the bonds between those base pairs.
So it's like unzipping a jacket.
Precisely. Then another enzyme called primose comes in and lays down a short RNA primer, like a starting block.
Okay.
And then comes DNA polymerase, the star builder,
Right.
adds nucleotides to the primer, meticulously matching them up with the original DNA strand to create a new strand.
Wow. It's like a microscopic construction crew working with incredible precision.
Absolutely.
But I've heard there's a leading and a lagging strand.
Yes.
Why is one built continuously
Hmm.
and the other kind of in pieces?
Yeah, that's because DNA polymerase can only build in one direction, from the 5-foot end to the 3-foot end.
Okay.
On the leading strand, it's smooth sailing.
Gotcha.
But on the lagging strand, it has to work in fragments. These are called Okazaki fragments.
Okay.
And those are later stitched together by another enzyme called DNA ligase.
It's like building a brick wall in reverse.
Yeah, that's a good way to think about it.
I never realized DNA replication was so intricate.
It's fascinating.
But, DNA is just the blueprint, right?
Right.
How does this code actually become something tangible, something that makes us, well, US?
That's where the central dogma of molecular biology comes in. DNA to RNA to protein. Think of it this way. DNA is like a master recipe book locked in the cell's nucleus.
Okay.
To make a protein, we first need to transcribe that recipe, copy it onto a note card.
Got it.
That note card is RNA.
Okay, so RNA is like the messenger. What makes it different from DNA?
So a few key things. RNA is single-stranded, not a double helix.
Okay,
It uses uracil, U, instead of thymine, T.
gotcha.
And most importantly, it can leave the nucleus, carrying DNA's instructions to the ribosomes, which are the protein factories in the cytoplasm.
So the ribosomes, that's where the action happens.
That's where all the action happens.
They're like little construction crews, right?
Yes.
Taking those RNA instructions and building, well, everything.
Exactly. Ribosomes are made of ribosomal RNA, rRNA, and proteins, and they read the messenger RNA, the mRNA code, which is divided into these 3-letter words called codons.
Okay.
Each codon specifies a particular amino acid.
So it's like a 3-letter code for each ingredient.
Yes.
And the ribosome just knows exactly which ingredient to grab based on that code.
It's like a cosmic takeout menu.
I love that.
Each 3-letter combination tells the ribosome which amino acid dish to bring. And just like a menu, there are specific codons for start and stop, telling the ribosome where to begin and end protein synthesis.
That's incredible. It's like a perfectly coordinated system, with each molecule playing a specific role.
Mhm.
I'm picturing those little tRNA molecules as delivery trucks,
Yes.
bringing building materials to the ribosomes, following those instructions on the mRNA menu.
That is a great analogy. And this whole process, from DNA to RNA to protein, it's happening constantly in your cells, orchestrating everything from your hair color to your digestion.
It's mind-blowing how much is going on at that microscopic level. But what happens when there's a typo in the DNA blueprint? You know, a mutation.
Yes.
Are they always bad?
That's a great question, and one we will definitely dive into next. Mutations are those changes in the DNA sequence.
Okay.
And while they can sometimes be harmful, they're also the engine of evolution. Without mutations, there would be no variation, no adaptation. It's a fascinating topic, and we'll uncover more about the different types of mutations and their impact in the next part of our deep dive.
I'm already hooked.
Awesome.
But before we get ahead of ourselves, let's stick with the basics for now. We've talked about DNA as this long, intricate blueprint. But how is all that genetic material actually organized within a cell? It must be packed in pretty tightly.
You're absolutely right. That's where chromosomes come in. They're like super organized filing cabinets for DNA.
Okay.
So imagine DNA as a long thread wrapping around proteins called histones, forming nucleosomes.
Okay.
These nucleosomes coil further into solenoids and then supercoils,
Wow.
packing all that DNA into a neat little chromosome.
So it's like taking a really long string and winding it up into a compact ball.
Exactly. And this packing isn't just about saving space. It also plays a crucial role in regulating which genes are active and which are silent.
Oh, so there's a reason for all this coiling and packing. Tell me more.
Absolutely. So euchromatin, the loosely packed form, is accessible for transcription, meaning genes can be read and used to make proteins.
Okay.
Heterochromatin, on the other hand, is tightly condensed, like a closed file, keeping those genes inactive.
Gotcha.
And fun fact, if you stretched out all the DNA in one of your cells, it would be about 5 feet long.
Wow, that's longer than I am. It's incredible how much information is packed into every cell. But cells don't just sit there, right?
No, they don't.
They divide, they multiply.
Exactly. Cell division is essential for growth, repair, and reproduction. And there are two main types, mitosis and meiosis.
Hold on, mitosis, meiosis, are these the same thing?
They have distinct roles.
Okay.
Think of mitosis as making photocopies, creating identical copies of a cell for growth and repair. It's a single division resulting in 2 daughter cells with the same number of chromosomes as the parent cell. Meiosis, on the other hand, is like shuffling a deck of cards,
Okay.
producing genetically diverse gametes, sperm, and egg cells for sexual reproduction.
Okay, so mitosis is for growth, meiosis is for making babies.
You got it. And we'll delve deeper into the fascinating details of both those processes in the next part of our deep dive.
It's really cool stuff.
It's really cool.
Yeah.
All right, we're back and ready to continue our deep dive into this amazing world of biology. Last time we left off talking about cell division, mitosis for growth and repair, meiosis for making those genetically diverse gametes.
Right, right.
But before we get into like the nitty-gritty of those processes, I'm still curious about mutations.
Okay.
We touched upon them briefly, but like what actually causes them in the first place?
Well, as you can imagine, replicating billions of base pairs of DNA with every cell division,
Yeah,
it isn't always perfect.
right.
Sometimes errors can slip through the cracks.
Well, a typo every now and then. I know I make plenty of those. Exactly. And these are what we call spontaneous mutations. They can happen during DNA replication.
Got it.
Remember DNA polymerase adding those nucleotides to the growing DNA strand?
Yeah.
Well, sometimes it adds the wrong one, like a mismatched puzzle piece.
And that's it. One wrong nucleotide and boom, a mutation.
It can be that simple, although DNA polymerase does have proofreading mechanisms to catch and fix most of those mistakes.
Right.
But occasionally some slip through.
Gotcha.
And these errors can involve substituting one base pair for another,
Okay.
inserting or deleting base pairs, or
Okay.
even affecting entire sections of chromosomes.
So those typos can happen at different scales, from like a single letter to a whole paragraph.
Yeah.
But what about those induced mutations that you mentioned? What causes those?
Those are caused by external factors, things that can damage DNA and increase the likelihood of errors during replication.
Okay, so like environmental factors. I've heard that UV radiation from the sun can damage DNA.
Exactly. UV radiation, X-rays, certain chemicals in cigarette smoke, even some viruses can damage DNA and lead to mutations.
Yikes. So we need to be careful about what we expose ourselves to.
Yeah.
But you mentioned that not all mutations are bad.
Right.
Some are even beneficial. How is that possible?
Well, that's a crucial point. Mutations are actually the raw material for evolution.
Okay.
Think about it. If our DNA never changed, there'd be no variation, no adaptation.
Right.
It's these random changes, even the harmful ones, that create a pool of genetic diversity that natural selection can act upon.
Well, so it's like a game of chance, with some mutations being beneficial, some harmful, and some having no effect at all. And
Exactly.
the beneficial ones are the ones that help organisms survive and reproduce better.
Precisely. It's this constant interplay between mutation and selection that drives the evolution of new traits and ultimately new species.
That's incredible. It makes you appreciate the power of these tiny changes in our DNA. But let's shift gears a bit. We've been talking a lot about how our cells work, but what about when they don't? We touched on cancer earlier, which is uncontrolled cell division. Can you explain how that actually happens at the cellular level?
Of course. At the heart of it is a disruption of the normal cell cycle,
Okay.
that carefully regulated process of growth and division. There are checkpoints throughout the cell cycle where the cell assesses its own health and integrity.
Hmm.
So if there's DNA damage or other problems, the cell cycle can be halted for repairs, or in some cases, the cell might even self-destruct to prevent the spread of that damage.
It's like a built-in quality control system.
Precisely.
Okay.
But in cancer, these checkpoints can be overridden.
Oh.
Mutations in genes that control the cell cycle can lead to uncontrolled growth and division.
Okay.
For example, a mutation in a tumor suppressor gene, which normally acts like a brake on cell division, can allow cells to proliferate unchecked.
So it's like the brake pedal stops working and the cell just keeps dividing.
That's a good analogy. And as these cells divide uncontrollably, they can form a tumor, a mass of abnormal cells.
Right.
If the tumor stays localized, it's considered benign.
Okay.
But if it invades surrounding tissues and spreads to other parts of the body, it's malignant, which is what we typically refer to as cancer.
That makes sense. So cancer is essentially a disease of the cell cycle caused by mutations that disrupt the normal checks and balances.
Exactly. And it's important to remember that cancer isn't a single disease, but rather a group of diseases with a wide range of causes, characteristics, and treatments.
That's a lot to take in. It makes you appreciate the complexity of our bodies and that delicate balance that keeps everything functioning properly.
Yeah.
But shifting gears again, remember that central dogma we talked about earlier,
Right.
DNA to RNA to protein.
Yes.
We discussed transcription briefly where DNA's code gets copied onto RNA.
Yeah.
But what are the specifics of that process? How does it actually work?
Well, just like DNA replication needs a primer to get started, transcription needs a promoter. Think of it like a signpost on the DNA strand that says, hey, RNA polymerase, start here.
RNA polymerase, is that like the DNA polymerase we talked about earlier?
Similar idea, but this one builds an RNA strand from the DNA template.
Okay.
It binds to the promoter, unwinds a little section of the DNA double helix, creating a transcription bubble, and starts linking RNA nucleotides based on the DNA sequence.
So it's like copying a recipe from the cookbook, DNA, onto a note card, RNA.
Exactly.
Only this time, RNA polymerase is the one doing the copying.
Precisely. And just like DNA polymerase has a leading and lagging strand, RNA polymerase can only transcribe in one direction.
Okay.
From the 3' end to the 5' end of the DNA template, meaning it builds the RNA transcript in the 5' to 3' direction.
Okay, I'm following so far. But if RNA polymerase is reading just one strand of the DNA, does it matter which strand? Are they both identical?
It absolutely matters. The strand that's being transcribed is called the template strand, or sometimes the antisense strand.
Got it.
The other strand, the one that's not being read, is called the coding strand, or the sense strand.
Okay.
It's called that because it has the same sequence as the RNA transcript, except with thymine T instead of uracil U.
Interesting. So it's like having a master copy and a working copy of the recipe.
Yes.
But you said earlier that this RNA gets processed before it leaves the nucleus. It's not ready to go straight to the ribosomes.
You're right. Especially in eukaryotes like us, there's a bit of editing that happens first. Think of it like revising a rough draft before sending it off for publication.
So what kind of edits are we talking about here?
Well, first, there's capping. A special cap gets added to the 5' end of the RNA transcript, like putting a protective helmet on it.
Okay.
This cap helps the mRNA bind to the ribosome and protects it from being degraded.
So it's like protecting the instructions from getting crumpled on the way to the construction site.
Exactly. Then there's the poly-A tail. A string of adenine nucleotides gets added to the 3' end, like a tail on a kite, which also helps with stability and export from the nucleus.
Okay, so we got a cap and a tail. What else?
Now comes a really interesting part. Splicing. You see, eukaryotic genes have these sections called introns and exons.
Right.
Exons are the coding regions, the parts that actually contain the recipe for the protein.
Okay.
But introns are like unnecessary interruptions. They need to be removed before the mRNA can be translated.
So it's like cutting out the fluff and
Oh,
keeping
what?
only the essential ingredients.
Precisely. And this splicing process is done by a complex called the spliceosome. It identifies the introns, cuts them out, and then stitches the exons together, creating a mature mRNA molecule that's ready for translation.
Wow, so it's like a cellular editing team making sure the final message is clear and concise. But wait, why even have introns in the first place? It seems like extra work for the cell.
That's a great question. Scientists are still unraveling all the mysteries of introns, but we know they play a role in regulating gene expression and can even lead to different versions of a protein from the same gene through a process called alternative splicing.
Alternative splicing. So it's like having multiple recipes from the same set of ingredients,
It-
depending on how you arrange them.
Precisely. It adds another layer of complexity and flexibility to gene expression.
This is getting more and more fascinating. But I think we've covered enough ground for now. Next time, we'll delve into the world of chromosomes, explore the intricacies of mitosis and meiosis, and even touch upon the revolutionary field of gene editing. Stay tuned.
Welcome back to our deep dive into this amazing world of biology. We've journeyed from the twisted ladder of DNA to that intricate dance of transcription where DNA's code gets transcribed into RNA.
And today we're going to zoom out a bit
Okay.
and look at the bigger picture, starting with those super organized filing cabinets of DNA, chromosomes.
Chromosomes, right? Those like X-shaped structures we've all seen in like biology textbooks. But have to admit, I still find them a bit mysterious. What exactly are they and why are they so important? So think of chromosomes as condensed packages of DNA.
Okay.
Remember how we talked about DNA wrapping around proteins called histones to form nucleosomes?
Yeah.
Well, those nucleosomes coil and supercoil further, packing all that genetic material into these compact structures we call chromosomes.
So it's like taking a really, really long thread and winding it up neatly so it fits into a tiny space.
That's a great analogy. And this packaging isn't just about saving space.
Okay.
It also plays a crucial role in ensuring that DNA is properly distributed during cell division.
Ah, right. Cell division. That's where mitosis and meiosis come in, those two processes we keep mentioning.
Exactly.
Can we finally delve into those now? I'm ready to learn the difference between making copies for growth and shuffling the deck for reproduction.
Absolutely. Let's start with mitosis, the process of creating identical copies of a cell for growth and repair.
Okay.
Imagine you have a single cell and you need to make two identical copies. Mitosis is how you do it.
Okay, so it's like making a photocopy of a cell.
Exactly. And it's a highly choreographed process with distinct stages. First, during prophase, the chromosomes condense and become visible, like dancers taking their positions on a stage.
Okay.
The nuclear membrane breaks down, and the spindle fibers, like a set of ropes, start forming.
So prophase is like setting the stage for the big dance.
Precisely. Next comes metaphase, where the chromosomes line up neatly at the center of the cell, attached to those spindle fibers. It's like the dancers forming a perfect line across the stage.
Okay, I'm picturing it. What happens next?
Then in anaphase, the sister chromatids, those identical copies of each chromosome, separate and move to opposite poles of the cell. It's like the dancers gracefully splitting and moving to opposite sides of the stage.
So each side of the cell gets a full set of chromosomes. That's brilliant.
Exactly. And finally, in telophase, the nuclear membrane reforms around each set of chromosomes.
Okay.
The chromosomes decondense and the cytoplasm divides, resulting in two identical daughter cells.
So it's like the curtains closing on each group of dancers, marking the end of the performance. Wow, what a perfectly coordinated dance. But what about meiosis? How does it differ from mitosis?
So meiosis is all about creating genetic diversity, which is essential for sexual reproduction.
Okay, right.
Remember those gametes, the sperm and egg cells?
Yeah,
They're produced through meiosis.
right. And they only have half the number of chromosomes as other cells.
Exactly. And that's because meiosis involves two rounds of division.
Okay.
The first round, meiosis thigh, is where the real shuffling happens. Homologous chromosomes, one from each parent, pair up and exchange genetic material in a process called crossing over.
So like the chromosomes are swapping pieces of information with each other.
Precisely. This shuffling creates new combinations of genes, which is why siblings from the same parents can be so different from each other.
Right.
Then these shuffled chromosomes separate, reducing the chromosome number by half.
Okay, so that's meiosis on Earth. What about meiosis the second?
Meiosis II is similar to mitosis. The sister chromatids separate, resulting in 4 haploid daughter cells, each with half the number of chromosomes as the original cell. These are the gametes.
So mitosis is like cloning, and meiosis is like shuffling and dealing a deck of cards to create unique combinations. I finally get it.
Excellent. And this intricate dance of cell division, from mitosis to meiosis, is happening constantly in your body, ensuring growth, repair, and the continuation of life itself.
It's truly awe-inspiring. But speaking of awe-inspiring, remember we touched upon gene editing earlier, that revolutionary technology that allows scientists to actually manipulate the very building blocks of life. I'm dying to know more about that.
Ah, yes, gene editing. It's a rapidly advancing field with incredible potential. One of the most powerful tools in this field is CRISPR-Cas9, which allows scientists to precisely edit genes, like changing a single letter in the DNA code.
Wait, so we can actually go in and fix those typos, those mutations we talked about?
In a way, yes. CRISPR utilizes a system that bacteria use to defend against viruses. It involves an enzyme called Cas9 that acts like molecular scissors, cutting DNA at a specific location guided by a piece of RNA.
So it's like a targeted search and replace for DNA.
Exactly.
Find the error, cut it out, and replace it with the correct sequence.
Exactly. And this technology has incredible potential for treating genetic diseases, developing new therapies, and even engineering crops with desirable traits.
It sounds like something straight out of science fiction.
It's amazing.
But with such power comes great responsibility, right? There must be ethical concerns surrounding gene editing.
Absolutely. The ability to edit our own genomes raises profound questions about safety, equity, and the very definition of what it means to be human. It's a powerful tool that requires careful consideration and responsible use.
It's like having a magic wand. Incredibly exciting, but also potentially dangerous if it falls into the wrong hands. Well, we've certainly covered a lot of ground in this deep dive, from the basic building blocks of DNA to the complexities of cell division, mutations, and even gene editing. It's been an incredible journey through that microscopic world that makes us.
Yes. And remember, this is just the tip of the iceberg.
Right.
There's still so much we're learning about genetics, cell biology, and the intricate processes that govern life itself.
It's truly humbling to realize how much we don't know and how much there is still to discover. I feel like I've gained a whole new appreciation for the complexity and beauty of life.
That's the beauty of science. It's a never-ending journey of discovery. And the more we learn, the more we realize how interconnected everything is, from the tiniest molecule to the grand scheme of evolution.
So next time I look in the mirror, I'll remember that I'm not just seeing a reflection, but a masterpiece of biological engineering, a product of billions of years of evolution, and a walking, talking testament to the power of DNA.
Beautifully said. And never stop asking questions and exploring the world around you, because there's always more to learn and marvel at.
Thanks for joining us on this deep dive into this fascinating world of biology. Until next time, keep exploring, keep learning, and keep marveling at the wonders of